It is Wednesday night, guys. We are back to Outside the Enclosure. Outside the Enclosure, one of the 3 that we're doing now. We've swapped it out from just No Remorph to include a couple others. And I'm going to be right up front. We kind of cheated already. We're only on episode 2, and we kind of cheated, and we went right back into reptiles with this. But you're not going to hear talk about collections and what we're pairing and how many clutches we're going to have. It's not, hey, how long have you been...
That.
Yes, it's going to be outside of that a little bit, right? Not a long ways, but somehow we talked Ben Morrill, Dr. Ben Morrill, into sitting down with us and explaining some of the confusion we hear in all the different chats we're in. Hey, how come we can't make this? Or I don't understand why I'm pairing this and I'm getting something that looks like this, but it's not testing right. All of the goofy genetic questions for those of us involved in this day to day. And a lot of the people that watch this every week aren't at all. So they have no clue what we're talking about when we say het or homozygous or, you know, this is allelic with that. Or, boy, I hope I get a super in this pairing, you know. They're lost on that. And they're like, all right, guys, your snakes look awesome, but I have no clue what you're saying. So this goes out to all of those people and all the confused ones in the chat that still say things that completely baffle me because I'm like, what are you talking about, man? So we're going to hopefully get some of that straight. Ben brought some slides. Obviously, he knows this stuff inside and out, but we told him, kick us around the little soccer pitch like we're 5 years old with this stuff and kind of beat us with some general terms.
Well, I think you said 12, not 5.
Well, I feel like I want to be on the 5 end. Was it 12? Well, I could be wrong. It's only 7 years, guys, and who knows, you know.
I think we go with 12. We don't want them coming out with crayons and stuff like that.
Right.
I'm thinking of my understanding of genetics and where that lays on the scale compared to a doctor, and I'm a 5-year-old in that realm. So, yeah, no offense to any of the 5-year-olds out there graduating from, you know, Harvard or whatever, summa cum laude. It's okay. You guys are different, but yeah. So the whole idea was, yeah, let's get it to basics. Let's understand it, and let's have a good time with it. So thank you to Rare Genetics Inc. out there and Ben for agreeing to do this. It's going to be a fun one. Let's say hi. Who all do we got out there? I see the numbers are pretty good. We got a lot of different faces in there. I see the comments rolling, so that's awesome. Ella, good to see you. And she's just one of those nerds kind of like us that's probably going to know all this and be like, guys, what are you doing? But
she's pretty excited about it.
Yeah, looking forward to this one.
Oh,
Sammy.
Nice, okay.
I'm sporting Sammy the bull.
Brand new sweatshirt from Sammy.
Yeah, yep.
And dude, can't get a nicer guy in the community, real honestly. But appreciate you, sir. Dickie Foster. Man, we need to clip Ken saying that. Dickie Foster.
Yeah, we do.
So excited.
That would be hilarious.
Tim, there's Dad. Another college professor that's going to be in a different class than he used to teach. This is going to be genetics for you, Dad. So also, guys, pop quiz at the end. Instead of trivia, we're calling it pop quiz tonight. Ben's putting on a class. We're going to quiz you for the $10 Forge coin at the end of the show. So appreciate everybody in here. Oh, we got Connie, the other side of the Tim Connie combo. Feeding each other beers, I'm sure, and they're going to learn about snake genetics tonight. We got G-Paw. Good to see you, sir. Appreciate you. One of the great affiliates in the group. Good to see him out here. Another good affiliate right there, Pacific Northwest Cookie, Maine. Baked goods shipped all over the country. If anyone's interested, shoot your Forge coin out there for a cupcake. Brave Adventures. Good to see you, sir.
It's got a new
Always
logo
in
there.
here. He does, yeah. He keeps swapping them up. Sometimes it's Star Wars, sometimes it's other things. Dwight, appreciate you, sir. Good to see you. Thanks for coming in tonight. Appreciate all the music through the week. Keeps things going. Nick, 666. Good to see you, sir. Always out there with the RWO crew and his Sipping with Serpents thing he does Saturday night. So if you can swing over there, guys, check that out. Dom, good to see you, sir. Haynes Gang. Always nice to see you in the house. Appreciate you coming out. We got E. I just call him E at this point, man. He's everywhere. Reptile dysfunction. We're not covering that topic tonight. That's a completely different field of study. I don't think Ben is up on reptile dysfunction, but maybe we could throw that out there. Mission Morphs. another affiliate. Appreciate you, sir. Thanks for swinging in. Fantastic giveaway last week, guys, with Mark Mandick on, and can't say enough good about that group, man. You
Go
guys
back
are,
and watch
you
it.
guys are awesome.
It's really
it was
good.
a really good
Yep.
show. Yeah, had a good time. Appreciate that outfit, though. John, Bridget, and Chance. Shed it out. Look at this. Jamie's buddy. Chris, thank you for
Yay.
swinging in. Hopefully things are going good. We got a show coming up with her. She's going to bring some geckos to an expo with us, and we're going to see if we can move some stuff over in Minneapolis, St. Paul. White Feather, Val, congrats on the 3-egg clutch and proving out those double hets. Well, triple hets that you got a couple double visuals out of. I should be
With
more
the
specific.
Silver Surfer.
Yeah, Silver Surfer went into that pairing. So congrats.
It's awesome, man.
Yeah.
Lowe's, good to see you in here. Appreciate you. Thanks for swinging in. A lot of names we're starting to see, every week too, and that's awesome.
Yeah.
Man. I'm glad people are liking it. So,
Yep.
Big Dan, South Florida affiliate right there. Fantastic puzzle stuff from Hypo DG puzzle stuff. He's doing a lot of DG clown stuff too. So, hit up Dan, man. Awesome animals and a fantastic guy. There's the wife on the Rivens Forge team. Love those affiliates over there. Good to see you out there, Karina. Thanks for swinging in. Jason Opel, that guy is just plain trouble. As long as you see him in this show, you're good to go. If you see him closer than that, run the other way. No, I'm just kidding. He's a good dude, but good to see him in here. Oh, Cuddle Time. Good to see Cuddle Time back.
Yeah.
Appreciate that.
Yeah, Melinda was here last week too.
Yep. Starting to see her pop around again. Always good to see Melinda.
Yeah.
Super great lady, man, and really great ethic for this and loves her animals. That's obvious. So, Seawalk, good to see you, sir. Thanks for swinging in. Good Lord, the more we put up here, the more numbers go up. So let's keep it rolling. Tara,
All right.
good to see you. We're going to have you
okay.
on and just do rats at some point. I saw you on
Yes.
another
Yeah.
show and covering your rats.
That'd be
Awesome
good.
show. So, Aurora,
Yep.
cheers. I agree. I'm going to go ahead and toast to that. Anytime somebody
Yeah.
says cheers, I'm going to have a sip of beer. Can't
have to
can't
twist
go a
my
whole
arm.
show. But yes, cheers. Thank you for swinging in. Mitzi, smiley heart. Man, this is getting a little bit wild out there. Thank you guys for swinging in. Everybody wants to learn about genetics.
Awesome.
I think they just want to see Ben, to be real honest. But
Yeah, probably.
either way, we're going to do it. Sunday, I assume that Sunday seal coating. If you need an asphalt topping on your pavement driveway, this is the guy to go to if you're in the, what do we want to call it, north central U.S. He's going to cover like 4 different states, but does a fantastic job, does it on time, and
Yeah.
with a smile on his face, believe it or not. So, Chris, salute
good
out to
job.
you, sir. Go check out Sunday Seal Coatings.
Awesome.
Plus, he's just a cool guy. He's going to love that on-the-spot commercial, by the way. He'll be like, you are a dumbass, John. Jordan, good to see you, sir. Thanks for swinging in. One of the Puff Treon crew over there with F&A. Appreciate seeing Jordan out here for sure. Oh, look at this guy, Jeffrey. Man, look out for some crazy leopard gecko projects, guys.
Yeah. he's
Jamie,
got-
you know better than I do. What's he calling them?
Oh, they're Solar Flare is the one that comes to my mind right now, but he has 3 going. I don't
Okay.
remember right off the top of my head the names. of
But
the
very
others, but-
specific, like
Yeah.
things he's putting
Yeah.
into those and line
Yep.
breeding hard for that. So
Yeah.
appreciate Jeffrey.
Really
for sure.
amazing
Really nice guy
geckos coming
besides.
up. Yep.
Almost wore my GTL shirt tonight, but I did not have a chance to change. So
Oh,
tomorrow or next week over.
Next week, Jeffrey. Paul, good to see you, sir. Thanks for swinging in. Large Marge Morphs over there. One of the original 15 Minutes of Lame winners. But Paul's a fantastic guy. Good sense of humor. And
yeah.
you got to catch him on the right day, though. I will say that. You got to catch him on there. He's pretty spicy, that one.
Oh,
It's
man.
like that spicy hatchling that wants to nip you when you open the tub. But,
Those are
enough
the
of that
ones that make
silliness.
me laugh.
right, exactly. It's the entertaining ones. But yeah, thank you everyone for swinging out. We're going to get to this as quick as possible, not leave Ben sitting back there with slides. Guys, we got slides, we got graphics, we got things to go along with this, and you got an expert in the field for 20 plus years. You
Yeah.
can't
Ask questions. Put them in the chat. We'll get them up.
Please do, yes. And especially if it's related to what we're covering right at that moment, we will try to address that. So, couple commercials, guys. We'll be right back after that. Hang with us, and we'll bring Ben right out here.
Hey folks, here at The Forge we prioritize promoting people from the community, especially where we see opportunity to share some valuable insights or products with our followers. That is why we are thrilled to have B&S Reptilia as a sponsor. Chris is a fantastic example of bringing value to the community, with years of experience keeping a variety of animals from ball pythons and blood pythons to boas or colubrids and more. Chris brings a wide breadth of knowledge and a collection of beautiful animals. Chris also carries Coco2go and The Chipper, two of the highest quality reptile bedding products on the market. If you want to learn more about any of these species, are looking to add to your collection, or in need of some top-notch substrate, please don't hesitate to reach out to Chris at B&S Reptilia. Chris is also a member of a growing community of breeders who accept Forge Coin, so don't forget to show them your Forge Coin.
Listen, everybody says they support disabled veterans, until it's time to actually do something. Last year, through Operation Deeds Not Words, the Fat Man Nation with Ring Line Morphs raised $7,200 for disabled veterans and first responders through Camp Freedom. And it proved something important. When this community actually pulls together, we can make a real difference. So this year we're doing it again with Operation Deeds Not Words 2. And our goal this year is $10,000. Same mission, same attitude, just bigger. Camp Freedom provides outdoor adventures like hunting and fishing and just time back in nature for disabled veterans and first responders. Getting them out of clinical settings and getting them back to what really matters in life again. If you want to help us reach that $10,000 goal this year, then head on over to thefatmannation.com and pick yourself up an Operation Deeds Not Words t-shirt or a beanie. Because talking about helping people is easy. Actually doing something about it is what this campaign is all about.
Absolutely, guys. Deeds Not Words. We're getting kind of closer. We've got just a little over a month and a week or so to go on that. Nathan, good to see you in there, sir. Appreciate you swinging in. Yeah, so not a ton of time, guys. They have a goal that they're actually getting fairly close to. I'd like to see them exceed that by a couple of thousand this year and really make that a big thing, because watching what that provides for those first responders and those, veterans that are receiving that gift, it's awesome, dude. The smiles and excitement on those guys when they can get back into nature and throw a line in the water and they don't have to do anything and struggle with getting there. Somebody is there to facilitate that. That's just amazing. Least we can do, guys. So if you got it, also Forge Coin can go towards that, guys. If you're holding Forge Coin, you want it to go to Deeds Not Words, let us know. We will swap that directly over. So appreciate them and appreciate Chris for that. Jamie, we're sending you backstage. You're going to do... So people make Jamie work tonight. Send in the questions, make her filter and star that stuff so we can bring it up at the appropriate time. Enough stalling. Jamie, we got to say bye to you. We'll see you in a bit for the pop quiz. Bye. Good luck back there. Hopefully those things are coming in left and right. But Jason and I, we got to say hi to an expert in the field. Dr. Ben, right? Dr. Ben Morrill. Thank you, sir, for taking time out of your obviously hectic schedule and sitting down with us tonight and running us through all of the things we're wrong about until we hear it from an expert. Right?
We shall see. I'm definitely very happy to be here. I love when there are people interested in learning about genetics or reptiles, and when it's both, I'm extra excited.
This is right up your alley. This is the thing you do. See all of the background. Can you run us through just a real quick thing about your education, your background, and how long you've really been at this and where your level of expertise should be admired? I mean, it's pretty impressive, honestly, when I read through it.
Yeah. I've been. It's funny. People ask me, why do you like snakes? How long? Like, what is it that I honestly don't remember. Just my whole life, I've loved snakes and animals in general. And it's probably similar to many of you. But when I was moving out of my parents' house, I found a journal from fourth grade where my teacher had us write 5 lines every day, and I was writing about catching garter snakes and things like that. So it's always been there. In high school, I decided to cold call. I think I was a junior in high school. I cold called the local university and just asked if there's a herpetologist I could talk to. And she gave me a name. I called him. He actually let me come in and see his lab and talk to some of his grad students. And that was Dr. Joe Mendelson, who's now the curator of reptiles at the Atlanta Zoo. And just super patient and really, really cool. He grew up being a skateboarder punk kid and decided to go into academics to do reptiles, and so he took me in and was patient with me. And so anyway, just always that interest has always been there. Once I was in Dr. Mendelson's lab, he helped me meet another person, Dr. Mike Frender, who's also done some reptile stuff, but does a lot of evolutionary biology stuff. And that's when I started doing PCR and sequencing and all that kind of stuff. So I've been doing PCR and sequencing and designing tests and that kind of stuff since 2002 when I was in Frender's lab. And
Okay.
I ran a sequencing machine way back then, a really old school one with big old giant gel and this polyacrylamide. You have to be careful not to get
Pull
on
the
you.
plate like, oh,
Yeah,
yeah.
you have to make sure that polyacrylamide doesn't get on you because it's a neurotoxin and yeah, all this fun stuff.
Oh,
They
that I
don't let
didn't know. Okay.
yeah, they don't let people punk kids do that anymore, I don't think.
Too
Right. A little
dangerous.
iffy. Yeah.
Yeah.
And then as a grad student, I was working with reproductive biology, reproductive genetics in an animal science department. So everyone around me was all working with chickens and beef and pigs, and I did quite a bit of work with not any poultry, but I did a bit of beef and pig work, and we did some modeling stuff with mice pre-implantation genetics and their embryology stuff. So yeah, been doing lots of nerdy stuff. But I actually the Sutherlands TSK, the snake keeper, they had moved from California to northern Utah, and I grew up in Utah. And as some of my original plans as a PhD student were kind of falling apart, I just pitched the idea, hey, I actually talked to Dan and Colette, and she had this giant stack. I mean, it was literally this tall of all handwritten records. She would keep track of the length, the width, and the weight of each individual egg and which one hatched first, second, so she could kind of look at
Wow.
correlations like, is it the biggest egg that hatches first or last, or is there no
That's
correlation?
crazy.
Like, I mean, it was funny while I was there. Dan was making fun of giving her a hard time. He's like, most people it takes them, maybe 5 minutes to set up a clutch. It takes you 45, so you got to write down everything.
Yeah.
I got everything, and so I spent literally about 3 months when I wasn't in class or doing stuff at the bench that I needed to do as a grad student. I was just inputting, just data input for like 3 months, putting all that stuff in. So my dissertation is actually based on 10 years of ball python breeding that TSK did. And so you can,
Wow.
on
Okay.
our website, we have a link to it. But if you just put my name in and ball python quantitative genetics, quantitative reproductive genetics, something like that, you'll find it. You can read as much of it as you want. Super nerd stuff, but it is all ball python breeding stuff. So
That's cool,
So
man.
cool.
yeah, and
Yeah,
then
I'll
I
Yeah.
definitely check
got
that
the
out.
PhD and moved across the country to Virginia. And good friend Sean, we had talked online about breeding pythons for years, since about the early 2000s. But we met actually at an October in person, finally October 2011, the Tinley show, and he
Okay.
said, how come I can, because he did reptiles, he also did birds and fish. He's like, how come I can pull a feather out and send it into a company and they'll tell me if it's a boy or a girl, but we can't do that with reptiles. And so that's,
Valid question, right?
yeah.
Yeah.
Like,
So a
yeah.
lot of people maybe haven't heard anything at all. You think this is just popping up, but we've been working at this slowly since 2011.
Yeah.
Okay.
When did you
Awesome.
earn your doctorate?
2011.
2011. Okay, so I just got to ask. This is probably off topic, but it's something I'm curious about because you talked about the gels and then using what was a neurotoxin at the time. So the evolution of that, I'm gonna guess that you're using microplates at this point, or is that not true?
Yeah, so they switched to capillaries. So
Okay.
instead of running on a giant gel, they switched to these little capillaries. It was enclosed and much safer. But yeah, now a lot of the most cutting edge or next generation types of sequencing are done on chips. Yeah.
But that all happened after you were done with school and all that, right? That was kind of a
While I was a PhD student, definitely moving to capillaries happened while I was like right after I did my first sequencing project. Most
oh
places were switching over to capillaries, so that's
yeah.
been around since like early 2000s.
Okay.
But yeah, I would say the first time I did chip stuff, I was a PhD student.
Oh, okay. Yeah, I wonder. I
Gotcha,
didn't know a
gotcha.
timeline. I remember reading about those and the speed enhancement. I mean, it just kind of made sense, right? Didn't fully understand it, but understood it enough to be like, okay, this is what people should be doing. And then when I looked at like the panel tests and stuff, I'm like, that's what they have to be doing, right? When you're doing
Yeah,
a
yeah,
panel, if you're doing that with gel, I mean, how long is that going to take? Seems like it
yeah,
would be forever.
yeah. That would be painful. I don't want
Yeah,
to go back to the old days. That
right.
Like we have a
Okay.
machine to do a PCR. Like during COVID, all the PCR COVID tests that got done, millions and millions and millions, hundreds of millions, billions, whatever it was.
Yeah.
But yeah, so you have this machine, you put the mix in there with the sample, and it just it changes temperature like 95 degrees and drops down to 70 and then goes to like 65, and it goes like these three temperatures. Well, before my time, thankfully I wasn't back this far, but probably mid to
Turn the
late
dial.
90s, they would have three different water baths, and the
Oh,
grad student
gotcha.
would be sitting there, swirl it for a minute at 90,
Yeah.
take it out, swirl at 60. Yeah, you got to sit there for like
Oh
two
man.
hours swirling.
Oh,
That
no.
was rough
I miss
times,
that.
man. Wow.
Let's not go back to that, right?
Right.
But you mentioned something kind of odd to, well, not odd to me, but you were kind of focusing in mammals instead of the poultry. I assume the poultry's got to be a far closer thing to the reptiles, at least in what you're looking for and the number of chromosomes at play and things like that. Um,
Yeah, it was
is it
really just the professor that I had as my major professor. He did stuff in mammals, so I would have happily done chicken or turkey. There's actually, I think in the state of Utah, they produce like 30 or 40 million turkey a year. There's a lot of turkey in Utah,
yeah,
so there's some
Yeah.
cool turkey projects that they were doing. But yeah, it just happened to be the person I was working with. He was a cattle and I guess mostly cattle, and then like I said, that early embryonic stage. That some of that stuff we did in mice.
gotcha, gotcha. So one of my questions, and this is probably a dumb question, but I do think maybe some other people are wondering. It's all biological material, cell material, things like that. DNA all breaks down the same. Is there a different process that makes mammals or something easier than reptiles or poultry, or is it identical no matter what you're isolating?
Like to get the DNA.
Yeah, to actually draw your sample and do analysis on that, is it easier like through a skin cell versus a shed, or is it all just identical? It's biological material breaks down the
Yeah,
same.
it definitely makes a difference. And if you branch outside of animals, so I had friends that were doing genetics, plant genetics, and they have a cell wall in plant cells. We don't
Okay,
have that in animal cells. So yeah, they had to do extra steps that we didn't have to because we don't have a cell wall to deal with. So, but yeah,
gotcha.
so like for us, um, if you take a swab, like inside the mouth of a snake, just because a human, if you do a cheek swab, that actually usually gives you pretty decent DNA. From snakes, if you do cheek swabs, the quality is just not great. Shed skin is actually pretty dang good as long as you get it pretty quickly after the snake sheds and then get it away from any source of water, humidity, or any of that. Um,
Okay.
and then even better is a scale clip. So I don't think we've ever had to do a rerun on any scale clips unless it was like a problem with that run. But yeah, scale clips are awesome. They're fresh, and if you're getting just the very outer edge of that ventral scale that's clear, it's kind of like, cutting your fingernail. It's just that very clear skin on the outside. After a couple sheds, you don't even see it. But yeah, that's awesome. That works really, really well. And then
Okay.
the most surprising thing was we were doing some turtle stuff too, and someone sent us in toenail clips, and those we got buckets of DNA. That was way better than doing
Very
sheds
cool.
or scale clips.
So I assume for monitors, things like that, same deal. Like if you could get a clip of one of their nails, you're golden.
That should
Okay,
work.
that's awesome.
Yeah,
Very cool.
and I mean, as long as it's a piece of skin that it's not really high humidity in there, if that's the only monitor you have in that cage, and you just find a piece of shed that's in a corner or whatever, and it's been dry, it's been there for a while, it probably will still run just fine. And really, the first thing we do when we get the piece of shed is we break it into smaller pieces. So having multiple pieces, I usually tell people if they're working with lizards, just enough shed to cover the top of a quarter, and it doesn't matter if it's one piece or 30 pieces. That all works.
Okay. Awesome, awesome. Well, now, before we get back into the no remorse side, and we'll try to stay outside the enclosure a little bit, you brought some materials with you. You are going to kind of run, because there's a lot of people in the names and things I'm seeing in the list there that are not familiar with this at all. And we discussed a little before the show when we say het or homozygous, or we say superform or allelic with this, their eyes roll and they go, I have no clue what the heck you're talking about, because it's not something they ever deal with, right? Or that's what we kind of want to clear up a lot of.
Yeah.
Oh, go ahead, Jay.
I was just going to say, and then terminology that's used specifically for ball pythons isn't necessarily genetically true, but we use it and we understand it, right? The meaning that we're using it for works. And some of that explanation too, because people read things and they're like, well, I've never heard that. Like the codom thing we talked about.
Codom's a big one,
Yeah.
yeah. And dominant even is really confused in
Yeah.
how this all operates. So Ben brought some stuff that's going to lay a
gonna
little
be fun.
groundwork, I guess. Yeah, I'm kind of getting ready here too. Ben, not to put you on the spot, but we're going to shift modes a little bit. I'm going to do one of these things. We're going to put you out here to kind of prep us for what we're about to get. I'll add this as well, but it's going to be, well, let's shift it. Let's do this. And when you want that highlighted and you really want to move, if you want to switch and swap between those, just holler at me and I'll make that happen. But
Sounds good. Well,
let's see
honestly,
what you got. Yeah.
knowing me, I probably won't even think to have things swapped. You'll probably just want to do it when it makes sense. I'm not good. That's why I have Kayla help me when we do my show. I'm not good thinking about that kind of stuff. I'm not much of a presenter. I'm definitely a science nerd. Animal nerd.
Okay. No, that's fine. We'll do our best to kind of make that make sense for everybody. All right.
Good. So yeah, I mean, we'll start off with that question. Testing for leopard geckos. Yeah, I would like to do that. There is a little bit of scientific literature about some leopard gecko morphs. So yes, we hope to have that eventually. And 2026, we're moving out into other species. So I don't think it'll be too much longer. We'll be able to get some lizards in there too. So
Nice.
so yeah, the some of the information that we had talked about as we're preparing for this show is to be able to kind of help people understand what we talk about in genetics as modes of inheritance. So ways in which something that's in the parent or parents, how that passes to the offspring. So you as someone that's breeding animals, you want to be able to predict, if I put these two animals together, this is what I'm going to get. And so understanding how something is inherited, that mode of inheritance, helps you to be able to choose, what animals you want to keep as your breeders, and then when you have breeders ready to go, which ones to pair together. And obviously there's still odds, there's still crazy things that can happen. That's kind of part of the fun, is you never really know for sure what you're going to get.
It's
There's
fun
still surprises.
sometimes, Ben. It's fun sometimes.
That's true.
Yeah.
I'm an optimist. I focus on that. Yeah, you're right. I've had my heart broken many times too. That's true.
Yeah.
Oh, I had probably the craziest clutch I ever had. Like just looking at probabilities. I had 10 babies, all from one ball python clutch, and every single one of them was female. None of them were Partho. So that was crazy.
Wow.
I got lucky. That was a happy one. I know people that have had it the other way, and that's not fun.
Yes, absolutely. And
Yeah.
astronomical odds for that to be on that kind of number. I mean,
Yeah, yeah,
yeah.
definitely crazy. So yeah, if you do want to pop it up, so that we talked a little bit about the codom thing. One question a lot of the time that we get is, we're sending results out to people, and we'll say, hey, your animal, you had it tested for leopard, it tested as het, and they're like, well, it's a incomplete dom or a codom or dom. They'll say one of those things like it can't be het. So the first
That's
thing
all
that
it
I
takes.
the first thing I want to make sure you understand is if you're talking about a trait that's controlled by one gene, one mutation, like leopard. So we know from lots and lots of breeding, there's one mutation that if you have one copy of it, it looks like a leopard. That's called heterozygous, and it doesn't matter like the recessive or incomplete dom or codom, whatever. Those are terms that we use to talk about how the animal looks, or plant for that matter, how it looks and how that correlates to the mode of inheritance. But really, what it boils down to is for leopard, when we do that test, you either have 0 copies, so it's negative, you have 1 copy of the leopard mutation, that makes it a het, or you have 2 copies, and that makes it, het means heterozygous, homozygous, or hom. I might just say hom some of the time. So homozygous just means 2 copies. So it's just fancy words. 0, negative, 1, het, 2, homozygous. So same thing if it's a piebald. If you have a normal looking animal, you don't know it's possibly het or not. You do that genetic test. If it says negative, that means you have 0 copies of the piebald mutation. If it says het or heterozygous, you have 1 copy of the mutation. Homozygous means you have 2 copies of the mutation. And that's really all it boils down to. You don't have to make it any more complicated than that. It's really simple. And then, like I said, if somebody wants to call it recessive or dominant or incomplete dominant, that's to help us as humans to be able to communicate how it looks on the outside. That's not really genetics anymore. That's to be able to communicate to other breeders how they can predict this specific trait is going to pass when they do breeding. So
So
that's,
what you're telling us, Ben, is those terms are all just made up for us on the visual side
Yeah.
for us to understand what we're saying. That has nothing to do with the mechanics of how this operates.
yeah,
Correct?
if you know a trait is just controlled by one, like height in humans, that's not just one gene and one mutation. There's a recent paper, there was 10,000 different mutations they talked about in that can control how tall or short you're going to be. But in this case, where we're talking about a trait that's controlled by one gene, one mutation in one gene, yeah, there's just 0 copies, 1 copy, or 2 copies. And that's because every animal that's produced by sexual reproduction, which our snakes are, most of the time, they're getting 1 chromosome from their mom for all 18 chromosomes, 1 chromosome from their dad for all 18. So you really only have the chance to get 0 copies if neither parent gives it to you, or you could get 1 copy from the mom or just 1 from the dad. And then if you get 1 from both mom and dad, you've got 2 copies. That's why the genetic test, that's really all we can tell you is 0, 1, or 2.
Yeah.
Perfect.
So
Perfect.
GPaw had a question up there that I would just comment on it. I think that's true until you get to what we call the dominant genes, which means you've got a visual, which, like Dr. Ben just said, would be your het in testing or your het genetically, and then you've got the super, which is going to be your homozygous. So that's where it gets a
Well,
little bit
and
weird, even on the visual part.
and why we made up terms like recessive, because some of these
Yeah.
don't show visually with the heterozygous form. So we need a term for that to understand that it becomes visual with both copies only, right?
Yeah.
Or at
For
least
what we
visual
call
to us
recessive,
easily. Yeah.
Yeah, so For sure, when we're talking about, so that like what we just talked about, that's just the genetics and genetic test side of it. It's extremely simple, zero, one, or two. But if you want to be able to communicate to other breeders, hey, I've been working with this trait for the last 10 years. This is how I see it, passing on to the offspring. That's where these terms like recessive, dominant, incomplete dominant, a lot of the times people will tell us, hey, I've got this new thing that popped up in my collection. If you guys design a test, can you tell me whether it's recessive or incomplete dominant? And I'm like, no, we can't. That's something that comes after. You've got to figure that out looking at both phenotype and genotype, how it looks and what the
Right.
genetics say. That's how you can figure out which of those terms works the best. Now, they're terms that we've come up with to help us communicate with other people. The trait, the leopard trait or the piebald trait, it doesn't know any difference. It doesn't care. And some of those are a little leaky. Some of them have like a marker. And
Right. Right.
sometimes, like with pieds, sometimes, not all the time, you can breed a het, one copy in one adult or one breeder, zero in the other. So you bring a het to a normal. Well, those babies, you could then look at them, flip them over, and be like, oh, I see this, these dark track marks. That's the het pied marker. The
Okay.
probability is half of those babies should get that piebald mutation because the breeder that has one copy of the piebald mutation, it has one copy that's normal, one copy that's het pied. So it will pass one or the other. So that's why you get 50% possible hets. And you can flip them over and that marker's there. And so some people say, well, is it really recessive if there's a marker? Well, that's where it becomes, we as breeders have to decide. For me personally, if I'm going to explain to someone the piebald mutation, I feel much more comfortable calling it recessive because there's some ball pythons that have those tracks that are not het pied, and
Yeah.
there are cases where het pieds don't have that marker. So it doesn't fit perfectly into that mold every single time. The biology doesn't know it's not supposed to have the tracks if it's not a het pied, So it's a little
Right.
leaky, but it does help us to be able to communicate to each other. And so some of the drama and butting heads is whether something is incomplete dominant or whether it's recessive. And that's not the genetic testing side of it. That's the people trying
Yes.
to communicate.
Gotcha. And let's be honest, I mean, in every animal, you're probably going to see some indicator with a het, but we just don't see it or don't identify it or don't
Yeah.
know that it's the 75th scale from the cloaca to the, you know what I mean? There probably is that in every animal, but it has to be obvious, right? To fall into that bucket of recessive, at
Yeah,
least in my
obvious,
mind.
always there.
And
Yeah.
that's
Yes.
the phenotype. That's when we're talking about the visual expression, right?
Yeah.
Okay.
So
Just
how
wanted to-
something looks is phenotype, and the
Yep.
genetics behind it is the genotype.
Gotcha.
Gotcha. Perfect. Okay.
And so this, I like to show this picture when we're having this conversation because a lot of people will have seen something like this in like a high school biology class or something. But I also like to cover that stuff up with the animals that we're all excited about. So we'll talk a little bit about these things, but we'll cover it up with ball pythons, make it more interesting.
Okay. I appreciate that. The
Yeah.
flower lost me, I'll be honest.
And so these, and I know just seeing Punnett squares, some people get all freaked out just seeing them. They don't even like seeing the Punnett squares, but when there's ball pythons on it, hopefully that helps. So in this case, we're talking about albino and refer to it as being recessive. So if you look at the top here, you've got an animal that's completely normal. Oh, whoops, sorry, I switched. Okay, so you've got an animal that's normal, that's got this big R, and if it's albino, it has two little r's. And if you breed those together, you get all heterozygous animals, right? Because this one can only give the normal copy, this one can only give the albino mutation copy. So all the babies will have one normal copy and one het albino or albino mutation copy. And so that's why it's got a big R and a little r. And so when you breed two of these together, two hets together, that's how you get this Punnett square. And so if you have a big R, little r here as the sire, and then another one over here is the dam, and this is what you get as the offspring. So this is your mode of inheritance as being recessive. Anything that has one or two big R's is going to look normal. If it has both little r's, it's going to be the visual of the trait you're talking about. So
So
here,
perfect.
yeah,
People that
3/4 of
aren't
them.
for, I was
What's
going to
that?
say, people that aren't familiar with this, though, if we're looking at this and we say, okay, so both copies coming down are from one parent, both copies coming from the left, and where those intersect are the contributions from
Yep.
each parent. Okay,
Yep. So this
so
is mom gives a big R, dad gives a big R, you get big R, big R.
gotcha.
Yep.
Okay. All right.
Yep.
So we know the functionality of the Punnett square, and we understand how that operates. And every time they're going to give what they can give from either side, or statistically, right? So
Yep.
okay,
Yeah. So
gotcha.
this parent can give either a big R or a little r. This parent can also give a big R or a little r. And that gives you 4 possible outcomes. Genotypically, or the genotype, the genetics behind it. And since the heterozygous ones look the same as the homozygous normal one, and the only one that has the trait we're talking about is a little r, or homozygous for that mutation, that's what makes it a recessive. So this normal look is dominant over this albino look. So you have to have both copies come from the parents. So this little r had to come from both the sire and from the dam for you to be able to see the trait in the offspring. That's what makes it recessive.
Perfect. Okay.
Yep.
I think everybody can grasp that idea. I think everybody had that in their mind, but still to see the contributions and how that works out to a 1 and 4, it's a great way to see it. So
So
appreciate that.
pop quiz. You guys think about this mid conversation. We'll have even more of a pop quiz at the end, but pop quiz from this diagram here. How do we come up with 66% pos hets? So think about that for a second. How do we come up with 66% pos hets when there's 4 different outcomes here?
Well, you've got 3 of those. Yeah, 3 of those are the pos hets,
Yep.
right?
So this one isn't a het. Visually, it tells you it's homozygous, right? So that drops out. So now you have 3 that look normal. How many of those 3 that look normal are actually het?
Right, 2 of the 3.
2 out of
So,
the 3. That's why if you breed a het to a het, any of them that don't get the trait are 66% pos hets.
Yeah.
yeah, makes perfect sense.
Till you get the shed tested.
Yeah.
Right,
And you can
until
know
you
for
send
sure.
it in.
Don't have to raise her up and wait till she gives you a clutch to find out.
Right. No, that's perfect though. So,
So
three tries at it, and 2 of the 3 are going to be according to the odds. So
yeah, odds
2
wise.
times a third is 66. Gotcha.
Yeah. So then if a trait is dominant, then that means that mutation, that phenotype or the look that you get, if it has that mutation. So like we said with the ball python, the normal look is dominant over the morph we're going for. That's why this morph is recessive. But if we talk about leopard, if you have 1 copy of the leopard mutation, you get a leopard. And if you have 2 copies of the leopard mutation, you also get a leopard. It looks the same. You can't tell them apart visually. And we always kind of assumed that's what was going on with leopard, but we didn't really know for sure until we could do genetic testing. And we have tested many homozygous leopards for people now. And so then they know if they breed that leopard to something, every single one of its offspring is going to get the leopard mutation from that breeding individual. So that's very cool. So that's why this is dominant, because you get in the same type of breeding a het leopard, so a big R, little r leopard to a big R, little r leopard, a het to a het. So it's 1 copy in one parent, 1 copy in the other parent, just like we just did with albino. So it's the same type of breeding. But since this mutation, that look is dominant over the normal look, then instead of getting 1 out of 4 like we did for the albinos, now we get 3 out of 4 that look like a leopard. And only 1 out of 4 looks normal, because all we need is 1 copy. Whether we get 1 copy of leopard or 2, it's going to look like a leopard. And that's why it's dominant. That trait, that look, dominates over the normal look. And so in this case, you could actually also refer to them, and they have not provided this in Morph Market yet, but I think they may. We haven't really worried about it in ball pythons, but some other reptile species we might. But you could actually have a button you could put up there if you weren't doing the genetic testing. You could call these 33% POS super.
Oh, okay.
So, because 1 out of 3 is possibly homozygous.
Yeah.
And so this happens like in boas with hypos. A lot of people will talk about possible super hypo, possible, and also in corn snakes. I think Tessera, there's 1 or 2 of them in corn snakes that are dominant mutations, and so it's hard to know for sure which ones are the supers. So in this case, you could actually list your animal as being a 33% POS super.
Yeah, and I mean
Gotcha.
it would make sense to do that, especially if you're not going to test them, right? I mean, if there's a chance you get a super, no different than what we were talking about with the 66%. Yeah, it could be het, right?
Yep.
So I'm spotting something, and maybe it's my eyes. My eyes are terrible, by the way. So what am I seeing? We've got, okay. So we've got male to the top and female to the left, but wouldn't we have 2 big R's on the female side? Or am I reading that wrong?
So what's happening here, and I didn't go over this, but it's like what we did with the albino. So here we have a big R. So this is a super leopard bred to a complete normal, little r. And so then we take those offspring and breed them together. So we have a big R, little r, and then over here we have big R,
gotcha.
little r.
Oh,
So
okay,
that way we're
gotcha.
breeding het to het, just like we did with the albinos.
Oh, okay. That's where
This is
I
laid
got
out
confused
exactly
on
the
it.
same.
Okay.
So,
That was
and
my
I
bad.
didn't do a good job of explaining it, but that's just to show you, like, if you do a het-to-het breeding with a recessive, you'll see it 1/4 of the time. Whereas if you do a het-to-het breeding with a dominant, you'll see it 3/4 of the time.
Sure. Gotcha. Okay. So we're using the offspring in both cases here.
Yeah.
Yeah.
Oh, okay. That makes more sense to me. And I apologize. A lot of the little type is very hard for me to see. I hope everyone else can read that.
Yeah.
I
So
think
pause it and blow it up or whatever you need to do.
Right. I had to, Ben. I'm sorry. Onward, sir. You're doing great.
Yeah, you bet. So incomplete dominant, what's going on there is we use pastel as an example. So once again, if we take here a pastel, or like we would say from the genetic test, it would tell you it's het for pastel or has 1 copy. So we have a big R, little r. If we breed 2 pastels together, and this is an incomplete dominant trait, then 1/4 of the time it's going to get the big R from both the mom and the dad. So it'll be a big R, which in this case we're calling the super pastel. It could get a big R from the dad and a little r from the mom, or the other way around, and be a het. So it would be a pastel just like the parents. So that happens half the time. Then the other 1/4 of the time, it can get little r, and just look normal. So with an incomplete dominant trait, with both the recessive and dominant, we had 2 different phenotypes or 2 different looks that we could get. And we either, with the recessive, we got the mutated look 1/4. With the dominant, we got the mutated look 3/4. Well, in this case, there's actually 3 different looks, 3 different outcomes. If you have 2 copies, it's the most drastic change of a pastel or a super pastel. If you only have 1 copy, it's kind of in the middle. And if you have 0 copies, then it just looks like the normal still. And so that's why it's incomplete dominant, because it's not like either one of them is completely dominant or recessive to the other. there is a middle ground. So you can actually see visually if there's 0 copies, 1 copy, or 2 copies.
So again, these are terms for us using them visually only. I mean, it has nothing to do with the mechanics of how these genes are passing or what they're doing to the other gene. It's
Nope,
not like
still
one
zero,
gene saying, "Hey, get out
0,
of the
1, or
way."
2.
Yeah,
Right, right.
exactly.
I would like to lobby, if you have any say out there, Ben, I would like to lobby for that bottom right square to be called the Forge square. It seems, it seems like we always land down there in that bottom right. But
You're weighted towards that.
for whatever reason, yes.
Just
Anyway,
out of curiosity, what kind of demand do you see for testing incomplete dominance?
And so it's funny. I think a lot of breeders, especially ones that have experience by eye, like, being able to ID things. If you just came into breeding ball pythons and, like wall testing was available, you probably have a lot different feel and frustration or acceptance towards testing than if you had already done visual IDing for many years. But it's really cool the people that have a lot of experience, like Justin from Kobelka, from the very beginning, he was very patient and kind, just like Dr. Joe Mendelson was for me as a young student. Justin's been very patient as I've been really excited and wanting to make this happen for reptile breeders. He's excellent and has spent a lot of time. He essentially is down there, in his snake room, IDing things for hours and hours every week, and has been for many years now. But the cool thing is, he definitely on, like a medium to high dollar ball python, he just tests everything we have tests for, and so it's really cool. First of all, to see how amazingly good he is at IDing by eye, but it's also interesting the phone calls and texts we send back and forth. He's like, that's not yellow belly. I thought that was for sure yellow belly, and we'll talk about it. And a year or two later, he'll do some breedings and know for sure what's going on. And one time he texts me pictures of this clutch, and there's a couple of blue-eyed Lucy's in there. He's like, "There's no blue-eyed Lucy in this pairing. What in the world happened?" And so, we did a blue-eyed Lucy panel on them and found out that they were super phantoms. And he's like, "Oh, you know what? I did breed two. They did come
Yeah,
in the
in
same
the
line."
complex.
And
Yeah.
he had kept back the nicest looking ones. Well, the nicest looking ones were the ones that had phantom also, and he just had no idea phantom was in there. So, it's kind of interesting. Like, for some people, if you feel like it's not going to help you, and that's totally fine. But I think if you're, especially like with Justin, where he's always pushing to have new combinations, that means he couldn't have ever seen it because no one's ever seen it.
Right.
And
Yeah.
so some of those combos, it does trick you. Like GHI hides in some things, like it can be
Yeah.
there and you'll have no idea it's there. And that's just one that I've bred a fair amount, and I got tricked. When we came out with the GHI test, I had a clutch, and I was pretty confident I had to have some GHI and super GHI. It was Champagne GHI and Champagne Super GHI, and then there were Het for Clown. And I thought I knew which one was which, and it was really the really light colored one was the Super GHI, not the darker one.
Oh,
And so
wow.
that was kind
Ah,
of crazy.
okay.
Yeah.
Opposite of what you think.
we
yeah.
have a red stripe clown male right now that is not just red stripe, and I have a very sneaking suspicion it's GHI as well. So
Yeah.
I think that red stripe GHI combination is extremely hard to weed out. So that shed's coming to you. We're going to send you that one.
Right
Since
on.
you brought that up, I definitely need to know. There's
Yeah.
things I do not want GHI in coming from him. So
Yeah. And
Good
then
point.
there's some that like pastel, we've got pastel up here. Like I've bred many pastel combos over the years. And when they're babies, sometimes it's super obvious, but after they're a year old, in some combos, there's absolutely no way I could tell
Yeah.
whether there's pastel in it or not. A lot of the time I would ask people, I was like, "Do you have a picture of them right out of the egg?" And a lot of the time they've got
Yeah.
that purple head and it's obvious,
Yeah.
but after 4 or 5 sheds, that's gone and you can't tell visually. Then there's some things like, whether it has spider or woma or something that you definitely want to make sure you don't make home, you don't breed 2 of them together.
Right.
So sometimes they hide in combos, it's sometimes it's hard to know for sure. Like if you have spider and pinstripe and, hurricane and this and that,
Yeah.
a spider could hide in
That's
there. You wouldn't even know it's there. So,
absolutely. That's kind of what I thought right off the bat as we're looking at this. I'm like, the testing, what's the demand? And I'm like, well, you start stacking some of this stuff and it can get pretty confusing, especially if you have a couple genes that maybe do a similar thing and you're just not seeing a really, definite expression of it. So yeah, I can definitely see that.
Yeah, we're getting
and honestly,
stacked enough, right?
Yeah.
Stacked enough where it's rough.
Oh yeah. And honestly, another part to that is, you guys, if you've been on forums and things like that, how often do you see picture of my snake? Hey, just got this from PetSmart. What is it?
What is it?
Right?
Yeah, so we
Good
want to be
luck.
able to help people like that. Like they got this, they love this snake, they think it's awesome. They're like, what is it? And they have no information at all about what the pairing was or anything. And so we need to have a test for pastel and for, all those things so that someone like that can just, buy a panel and be like, hey, I got tested for these 50 things and I know it's this and this. I know it's not that, So a lot of the time, those incomplete dominant tests that you as a breeder, maybe you would never choose to use them because it's obvious to you. And especially if you have experience with those combos, I completely understand. You don't want to pay the money for that. But, we have a lot of people, and if you are a breeder, one of the most powerful things in helping you be successful is new people coming to the hobby. The more new people that come in that have excitement and are purchasing and learning and growing and bringing other people. And so that's another reason why I personally believe that's really powerful and really important is like someone just right off the street that just has one snake, they want to buy that panel and they want to have, know some of the things that's obvious to many of us. They're not going to have a clue, and we want to help them be able to know and understand and be able to communicate that.
Sure.
100%. Yeah, it makes total sense.
Well, and let's be obvious. I mean, the safeguard side of it too, for anyone selling an animal or receiving an animal to know that you're not just taking someone's word, hey, I see this in there. You've literally got like a sheet that says no, and it's not a question of your ability to see it or not, whether it's there. Like it's a confirmed genetic thing that you don't have that argument down the road. Like I tested for it, didn't show to be in there. I'm sorry you got that result or whatever. It's a safeguard for both sides, really.
Yeah, and we did, right when the testing started, we did have 2 or 3 people that had, and their names that most of you would recognize, but they had done like $20,000 or $30,000 deals with one person that I think was either getting mostly out or completely out. And then the testing was available and they sent it, and he had not been honest about either he'd made a lot of mistakes or not been honest or some of both. But yeah, that's another nice thing to it is if I spend $3,000 on an animal that I want to use as a breeder, the first thing I'm going to do is, even if I completely trust that person and they've already tested it. I mean, me personally, if I'm going to spend that much on an animal to be a breeder, I'm going to pay, $100 or $150 or whatever I feel like I need to do to get all the tests back. And, that way you can know right away if you really have what's going on. And I know it's happened with some bigger breeders where they probably, the picture they had on there did match the genetics. And then the animal that you get when you order, it's, one of the employees that packages it up, and it's not actually the same animal and doesn't have the same hets that you paid for. It's not something that happens very often, but if you're spending a chunk of money, you want to double check.
Yes.
Yeah. Well, just your time investment too. If you got something going into a project and you don't know for sure, how much time can you waste, right? You can waste a lot. Yeah.
Man, this is such an easy one to just get off the Nile
is.
into so many tributaries. Pretty quick, we're out in the Pacific. You know what I mean? It's so easy to do. All right, so what do we got here?
I think Kayla put these in just as pretty pictures. And that's it. So that's part of what we had presented before. So yeah, that's what I have for slides, and we can come back to them if we need to refer to them. But hopefully that helps connect some dots, and then you can come back to this and pause on a slide if you want to look at it at the Punnett square that has the animals you like instead of snapdragons or something else.
Right? So I think one of the big things we touched on, though, is the terminology thing that or at least in my mind, before I truly understood it, I assumed that the mechanic of the gene was somehow in the name. So when I heard dominant gene, I felt like in a equal pairing, that one more often than not will show up, and it has nothing to do with it. It doesn't say that the pairing is going to prefer that gene. It's just that's the one that's visually powerful over the others in the same odds. Right?
Well, like if you think about albino and leopard, those examples we had, um, what is dominant or recessive is the two different alleles. So at the albino locus, which is that location or the specific spot where that gene is, if you have normal genetics there, that's one allele, that's one set of instructions to make that normal protein. The mutated one, that's the albino one, that's the other, it's allelic, it's the other copy, the other allele. And what we're saying when we call albino recessive, we're saying that when it has that albino mutation, it's recessive in showing in the offspring. So it's not as powerful as the normal genetics. Whereas
Gotcha.
leopard, on the other hand, if you have one allele has the leopard mutation, one is normal, that is dominant over the normal look. So you'll actually see it even if you only have one copy. So when people are saying dominant or recessive, you're talking about how the alleles interact with each other, not how genes interact with each other.
Absolutely. Gotcha. Yeah, so I think that covers the base mechanics of how things operate, but I know there's a ton of questions out there, and we talked a little bit before the show on things like lines of a particular. Again, we're going to use this touchy because I think it's confused, but red stripe type, and well, let's just use red stripe because I think there's a lot of animals out there that are presenting like a red stripe, but not meeting the genetic requirement to test as a red stripe. They may look identical visually to our eyes and the things it does with the pattern and a little bit of a color tint that it brings in, but on the test it's not coming up as red stripes. So when we test for things, are we purely taking that example animal and the set of genes that are testing that way and assuming that's creating the color, or are we testing for what's actually creating the color and pattern?
I think there's
Does that make
an
sense? I mean,
assumption. There's an assumption in the beginning until
right?
something proves otherwise, right? And that kind of the direction it goes, like the desert ghost deal, right? You
Yeah,
assume,
yeah.
okay, this is it. Well, and now we're seeing this too, right?
So this might be a good way to explain it. So in the publication, I think this is from like 2022 or so, where they did the first work on albino and ultramel and lavender albino. This is Dr. Sedel's group at Eastern Michigan and students, which is super cool that she does that.
Right.
But yeah, so as they're sequencing, so they get, let's say it's just a round number. I don't remember the for sure numbers. Let's say there's 50 albino ball pythons. And so they sequence a gene that they know causes albino in other species, and they, oh yeah, we see a mutation in this TYR gene, and so it makes sense that this is likely what's causing it. But as they're testing all of those albinos for that new mutation they found, some of them were only testing het. And some of them actually tested negative, even though they can see they're definitely albinos. They look just like the ones that tested homozygous or two copies, but those are either testing het or they're testing negative. And so they had to keep sequencing. They went through the ones that tested negative, and they sequenced some more, and they found out, oh, somewhere else in the gene. So let's say that TYR, let's say it's 10,000 base pairs, so 10,000 A's, T's, C's, and G's. Once again, that's just a nice round number. It's not actual.
Right.
So we've got 10,000 A's, T's, C's, and G's. Well, if you make a mutation, say at number 732, a certain kind of mutation that causes a stop codon, that can give you an albino. But you could also do that at, 3,722. So our genetic test, that first test they came up with that was testing the first one, doesn't tell them anything about the other mutation that also can make an albino until they can find that second mutation. They don't know, they can't test for all albinos. They can only test for the ones that have the mutation they have a test for, because that test is very specific to that one specific spot. So then
Gotcha.
now if you think about axanthics in ball pythons, we know there's at least 3 different genes that are on 3 different chromosomes that if you have a mutation there can make it look like an axanthic, and there's enough variation in those 3 different lines that it's hard to know for sure just by looking at it. They look very similar. And so in that case, they had some axanthics early on, and then someone else found one. They're like, oh, we'll breed them together and make more axanthics, and then they got all normals. And then they found out eventually, oh, they're double het. They're heterozygous for 2 different lines of axanthic, so they look normal, but they are carriers for both those
Both lines. That's crazy.
So that's another possibility. We don't know for sure yet. That one possibility is there's another mutation or mutations for red stripe that we haven't been able to find. And some mutations are super easy for us to find. Some of them are more structural in nature and aren't like obvious. And we may have gone over it 70 times. We just didn't realize that that's what was making it look like a red stripe. So it could be that there's 5 different mutations in that same area, that same gene or whatever, that can make a red stripe, but we only have a test for one of them. And so if you happen to have a red stripe that has that mutation, our test will work. But if you have one that has a mutation somewhere else in the gene, or if there's multiple genes that do it like axanthic and it's in a completely different place, our test isn't going to work. And that's why we have a test grading system. And someone was bringing that up in the chat asking about why we have a test grade. So we still, the answer you get is still going to be 0, 1, or 2. It's going to be negative, heterozygous, or homozygous. But the reason
But
why
are we in the right place? Are we putting our focus on that test in the exact spot? That's the question, right?
And like when we run albino, we actually have to do 2 separate tests because we know there's 2 different mutations. Fire's the same way. We have to do 2 different tests. Ultramel, you get one result, but we had to run multiple tests for it. So
Even
with
in
red stripe,
compatible, though, even when they're compatible. So it's a package of genes or chromosomes that are
it's
creating
one gene.
that.
One gene, but it's 2 different mutations in that same gene.
Gotcha.
Yeah.
Okay.
So if you think about it, like if you have a paragraph that explains how to change the oil on your car, if you take out some of the words at the beginning or switch them, say do this or change it to do not do this, you can make it so someone can't change their oil. But you could also do that towards the end of the paragraph and completely mess it up. It's the same thing with that gene. It has 10,000 A's, T's, C's, and G's that give the instructions to make that TYR, if we're talking about albino, to make that tyrosinase protein. And you can mess it up anywhere along that way. That genetic test is only going to test for one spot.
Yeah.
And so if there's another mutation somewhere else that the genetics company doesn't know about yet, then it could slip through and actually have that trait and be heterozygous for it or homozygous for it, but our test is going to be blind to it. And so that's why our rating system, if it says it like red stripe, I believe is a D, rated D,
Yeah, I would
telling
think,
you,
right down
that's
there.
telling you, hey, if it comes out and says that it is a red stripe, it has 1 copy of the red stripe mutation, that should be correct, and that's good, and it matches what our test can do.
It's the
But
false
what
negative.
what we're letting you know is that we are aware there are other animals that look like red stripes that our test does not cover. As soon as we have it so that we can cover all of them like we've been able to do with albino and ultramel and all of those others, then that will fix that problem and we'll bump that grade up. And people that have paid for that red stripe test will be able to just send it in and say, hey, you did this for me in 2023 when you didn't have it. You just send it in with the same order number and we'll run it for you for free once we have it.
Oh, that's
That's
awesome.
an awesome feature.
Yeah.
So, okay, that's really good to know. So I did not know that about the grade. So great question from the
Yes.
audience, and thank you for explaining that.
And so if
I
it's a
didn't
grade
know-
A, we've put in a lot of time and we've run a whole bunch of known samples, and we know, like, I think that the number we have set is over 1,000. If we have 1,000, because a lot of the time the panels we run, people will let us know, hey, this is a clown het pied, pos het DG, pastel. So we know, okay, we ran a clown test on this and it matched what the customer gave us, check. So that's one. And after we've done 1,000 of those and they all match the customer's explanation, then that's when it can finally be an A grade test. So if you go to order a test from us and it's for a Morph you haven't ordered before, maybe you're ordering Phantom. You're like, I wonder how good of a test that is. And you look it up, you're like, oh, it's an A. Okay, great,
Yeah, a little more confidence in it.
Yes.
So can we talk about the flip side? Have you pulled tests that the people aren't seeing visually and they're still showing as a positive on your side? As far as like an incomplete or a...
Let's see. I think...
There has to be that case in there, I would think.
Yeah, so
with all of these tests.
Yeah, I mean, weird stuff definitely happens. I would say out of 100, there's usually 1 or 2 where we get a result that doesn't match, and it usually boils down to either we made a mistake or the customer made a mistake. An example of that was this customer sent me a picture. It was to test for clown, and she sent me a picture of a pied. She's like, it's pied faucet clown, but you just told me it's a clown. I'm like, well, it tested as being clown, and I can look at it. Yeah, that's clearly just a pied. It's not a clown. Well, after having her resend, shed, like all this stuff, we figured out she had put a clown het pied in to breed, and she had just pulled a piece out of the bin and sent it in, and it was actually from the male.
That's the tough part about.
Yeah.
So the field that I'm in, we have to list like uncertainty with everything. What's the uncertainty in your measurement, right? And for this specifically, it's almost impossible because you've got a variable. Well, I guess you could estimate it, but the variable of what happens with that shed before it ever gets to you is like, who knows, man. It could be anywhere.
Out of our hands. And obviously, if someone sends us the wrong shed, then they're going to get results that don't match what they expected. So there's absolutely nothing we can do to fix that. And then sometimes it's a problem on our side, especially when we first started, where we were manually making all calls and entering everything in. Now we have some of those things automated so that some of those problems went away. But sometimes, the person would say, hey, you said this is a DG, but it's not, like, it doesn't look like a DG. And we go back to the raw data. We're like, oh yeah, sorry, we entered that in wrong. And that still, once again, that wasn't very often. Like I said, it really is pretty consistent. Out of 100, there's going to be 1 or 2. And it's pretty much 50-50 whether the customer made a mistake or we did. So those ones, we'll have the person send another shed in, and then we test. We're like, oh yeah, now it is a het DG. It's not DG, and that's because it could be we entered it in wrong. It could be that we mixed up a shed. Could be that you mixed up the shed. All of those things are possible. So, So if you want a couple of examples, well, I guess I can really only give you one. That we really tested to death. So that we did do a video on this, but it was an animal that very clearly visually was a clown, visual clown, and it looked like it was a super inchy clown. And so the shed came in, we tested it, tested het for clown, and they're like, oh, something's wrong. So we retested the shed we had. We're like, it still says het clown. Like, can you send another shed? They sent another shed. It still said het clown. So I said, all right, send another shed. We ran it again. It's still, so we did a video on that, and just kind of, we had, I think we had Tommy on that from Inky Clouds, and who else? We had Dr. Travis Wyman also, and just kind of talked about, what in the world could be happening. But the other thing, and I mean, I still don't know for sure why that one won't test properly. That's a weird one. But that's, we're definitely tens of thousands, and I would imagine we're into the hundred or a couple hundred, or I don't know how many thousands of clown tests we've done. So that's extremely rare. But yeah, weird stuff happens. The other thing we've learned, and we have a cool video on this too, paradox animals. If you see patches of piebald, patches of clown, then patches that
Tests
aren't,
different.
they do, and they don't always match what they are visually. So we had Kenny from Bomb City. He sent a shed where he had marked before the shed came off. He had marked with a Sharpie 13 different patches. And
Yeah.
this animal had pied and sunset and DG in the breeding. And some
Yeah.
patches looked normal, some looked pied, some looked sunset. And then some
Yeah.
were either going to test het DG or normal. And so, patches obviously are pied, obviously for sunset. Sometimes they tested homozygous for pied, sometimes homozygous for sunset, but sometimes they didn't. And so even if you have a little bit of paradox somewhere on that animal, it may make it so the test is really wonky. So
That is
that's
super bad news.
Yeah,
Yeah.
I was gonna say we're not a gotcha channel and we're not a
Yeah.
gotcha podcast, but Ben, we just sent in, and I'm gonna remove this, but we just sent in a panel test to you because we have a nightmare animal.
more shed
we
than
have a
we do.
nightmare animal here.
and it's paradox.
It has, you name it in this thing. It's clown, it's desert ghost, it's hypo, it's black pastel, it's OD, it's acid, it's all of the above in there
Very
in a
cool.
different section, and we have no idea what this thing is.
It should not. It should not come back as homozygous for clown. But,
No.
but we
Well,
suspect
it
that
could.
it will.
I mean, it could.
Yeah.
But we think it's going to. We've had some testing done on it, and we just said, well, you know what, we're just going to do a panel on this thing. So I apologize, not to be
It'll be a fun one.
instigators, but I'm glad you went this direction with exactly where you went, because we have that test on its way, and I think you did put out a receipt. Hey, we got the test. It's underway. But I'm super curious to see what comes out of that nightmare.
Yeah.
I've never seen anything quite like it. But
That is awesome.
yeah,
That's pretty
and
cool.
clearly visually to us it looked hypo DG with all of the dark stuff in there. It's not just hypo, it's just way too light. I mean, that is a
Yeah.
very strange animal. But it comes
once
back
it-
as like not even het hypo. So
Yeah, when it
I
shed.
don't know.
So the first shed, when you looked at it, you'd swear hypo. You couldn't see any pattern. But this last one, when I pulled the shed out, you could definitely see it. So I'm going. It's not hypo, but it's just there's so much that it could be, and we threw around that whole chimera idea and then the androgenesis idea, and
Yeah.
we'll just be curious to see what you get for results on that one.
The thing that we haven't gotten yet, which they have seen like in birds and some mammals also, but for mosaics or well, I guess it'd have to be chimera. For chimeras, you actually can have parts of it that will test male and parts that will test female.
Oh, okay.
We haven't had that yet in a reptile that I know of. I know we haven't tested one yet. It will happen eventually.
Yeah.
There's, weird stuff happens. Like we were talking about earlier, you can know your ratios and all this stuff, and then just something pops out. You're like, what in the world?
Yeah.
right. No, 100% understand that. So that was my thing is like, is there a way to know parentage? Like, is there a way to know that it's andro or something like that both came from maternal or paternal?
Yeah.
'Cause I know
yeah.
some don't survive at all, but others kind of survive. They just won't reproduce.
Yeah.
You can test for that. Huh?
Yeah, if your question is just is this either andro or partho, if we get a shed from the dam and then from any potential sires, at least for sure the ones that were with her that season, and then it's kind of good if we can have others as well, just to make sure. But yeah, if we at least get the dam sire, or at least any sires that were with her that season, and then the sample from the individual you're interested, whether it's Partho or Andro, yeah, we can give you essentially, we give you a ratio. It's called the level of heterozygosity. And so from sexual reproduction, we know, like, kind of that range of percent heterozygosity that we can expect. And we find that when we run it on a Partho or an Andro, it's like at least a tenth, if not even below a tenth of what we would see in a sexually reproduced animal. The ones that we know from if it was a het albino female bred to a pied, and then you got some albinos out, we know it had to have been Partho. And so when we test those, we can see that level of heterozygosity drops quite a bit. So
Okay.
you could academically argue that maybe that isn't a perfect, proof. It's 100% proof, but it's pretty dang good evidence that that's a
Yeah.
Partho or an Andro.
Yeah, I didn't even know that existed. That's cool.
I didn't either, in parentage like that, who's your daddy, Maury Povich style thing. I didn't know if that was
Yeah.
part
We
of
can definitely
testing
do
that's
that.
available. Okay.
Yeah,
Perfect.
yeah. It's not something that we offer like through Morph Market, or you'd have to. It's kind of like more of a custom thing right now, but that will likely change by the fall, where you would be able to have that level of a test done. Like, instead of having a panel where we just tell you the 50 genes or whatever, we'll be able to do a panel and be able to have a identity for that individual animal and potentially have it registered, and you'd be able to track lineage and compare lineage and do relatedness if you're trying to decide, like, some species, like dry mark on the indigo snakes. The breeders are really worried about maintaining as much genetic diversity as possible. So we'll be able to, and they've been asking for this, and it's taken a lot longer than I hoped, but you'd be able to, if you have like 2 different males and 3 different females, you'd be able to look at that at a very, very powerful level, like p-values 0.000, like they do for forensics and stuff. You'd be
Yeah.
able to know, okay,
Awesome.
this male to this female will give you the best level of heterozygosity that you could possibly get, and there would be no
Wow.
question. For sure.
Yeah.
It's based on millions of markers to date in reptiles, which is how all breeding was. It used to be based on maybe a dozen or maybe a couple dozen markers that if you were able to do some kind of test. But this would be based on a whole genome, where we've got millions of markers in the ball python, and we're getting more and more in other species too. So, and then the
That's
other thing that
awesome.
would allow you to do is, say you do that panel on that animal and it's registered with us, and then we come out with the mahogany test in a year or 6 months, or maybe hopefully a lot sooner than that.
You don't have to rerun it, right?
Yeah, instead of paying for a new test, sending in another shed, you just say, hey, this is registered number blah, 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 blah. And I know you just came out with a mahogany test. Can you run it? And there'll probably be a way you can just go in, pick that animal, and say, hey, I want to update it for any new tests, or I specifically want mahogany done, and hit go. And it's going to be able to just go into that data that we have for your animal. And now it knows where to look for mahogany, where
Yeah.
when you first did it, we didn't know where to look. Now we
Sweet.
know where to
Really
look.
cool.
So,
So
Yeah.
full sequencing, really. I mean,
yeah.
pretty much.
Yeah. At some point, you'd almost have to sell those as, like, do you want us to retain it and just feed you future test results, or do you
Yeah.
want us to test this and throw it away?
So, like,
Right? I mean,
23andMe and like the dog,
yeah.
breeding ones, what they essentially do is, over time they'll have updates and be like, hey, we updated, this breed, that breed, or this disease, or this or that. So essentially we want to get to that point where we'd be like, hey, this last 6 months we updated all these things, and it could either be a subscription and you would just automatically get all updates. Anything you have registered, you get all that information, or you have the choice to pay $20 and then you would get all of your
Right.
tests upgraded or whatever. I don't know for sure how it'll shake out, but we should have that capability
Super.
by the end of the
Super
year.
cool, man. Yeah.
Yeah.
That would be really nice. Just the savings and time and stuff, and man.
Yep.
Heck
And then
yeah.
that also opens us up to be able to say, we reach out to breeders and be like, all right, you've got 20 females registered with us. if you can give us their clutch sizes, any breedings you've done with them and what their clutch sizes were, and we do that for 50 or 100 breeders or whatever, they get back to us, give us information, then we can find markers that increase clutch size. We could find markers that have to do with like the adult size of the female. If you wanted to select for larger females or smaller females or whatever you want to do,
Right.
that's the kind of stuff like I was doing as a PhD student for cattle and things like that.
Yeah.
Higher milk yield or better marbling or all those kinds of things. So once we start to have all that information on many animals, then we can reach out to people that have animals in our database and we can answer other questions. People that are into retics, we could find markers
Super
that say,
dwarf.
hey,
Yeah.
this retic has a 80% chance of staying under 10 feet or whatever. We could essentially give you something like they give for like dog breeds. It's 20% of this, that, we'd be able to do that for any questions we have. People always ask, can you test for Volta? I'm like, yeah, I mean, if we get them in the
If we get
database,
enough Volta in
Yeah.
here, yeah,
yep,
yeah.
if we have, a few that we know are for sure that, but the easier one to do is just reach out to people. Hey, we've got, 50 animals from you. If you could give us their weight or their length or both, then we can find markers for weight, length, whatever it is that we are interested in. We have that ability once we have that data there. So
So
Nice,
cool, man.
nice. Jason, you got Ben for a second. This is a first on No or Morph, but I got to use the restroom. I don't know if this has ever happened on the show before, but I apologize. I'll be right back,
you
guys.
gotta go, you
Jason
gotta go.
and Ben.
Yep. See you, man. That is so funny. I think that is maybe the first on the show, but something I was going to ask you about, and I didn't want to interrupt, so I let it go, and I'm trying to get back to what it was because it kind of tied in when we were talking. It was about you had mentioned like loci, right? And how does that? So when we talk about that, we talk about allelic, we talk about loci, and then we talk about. So you're talking about, well, it's in the same gene. It's just one of the A, T, C, right? So when we say loci, is that more? Are we narrowing in more into the gene, the actual positions in that gene?
Yeah, so
Okay.
locus is the singular, loci would be plural, and it's
Okay.
just a way to say location. So if I say the locus for cooking cookies is on page 72. I mean, it's the same kind of a thing. So if someone says locus or loci, they're just saying in this in this location. That's all that's. So you could say, this is the piebald gene locus or location of where the piebald gene is, or you could say the locus specifically for that mutation is number 732 of the piebald gene transcript or whatever. So it can be more broad. It could be the location of a whole gene, or it could be more specific where you say I want to communicate I'm talking about the
Okay.
location of a specific T changing to a C that makes
Gotcha.
it
So
a piebald.
it's a universal term. It just really matters the context, right?
Yeah,
Um,
yeah.
But then how does the allelic play in when we're thinking about a gene? Are we still talking about a location?
Yeah, so it's when you talk about something being allelic, what you're communicating to me, that's you're saying there are at least two different copies, two different like sets of instructions.
Mhm.
And so it could be as simple as one allele, everything's the same except for right here there's a T, whereas everything else here is the same, and right here there's an A. And like that like piebald is that way. There's just a single base change
And
that
they're
makes it
related.
so it can be a piebald. But yeah, it's in the exact same location, that A and T, the exact same part of that piebald gene. They're, base number 732 or whatever. And
Right.
then if that's a T, that's normal. If it's an A, it's a pied. And so
Gotcha.
so that's what allelic, what you're really saying is these are two different sets of instructions that are for the same thing, but there is a difference to them. If they were completely identical, I wouldn't call it a different allele. It's just one thing, one set of instructions.
Right.
But as soon as there's a change made to one of them, then we say that's allelic. So that set of instructions, it's to make the same thing. It's just there's at least one change there. And it could be several changes. You could say these are allelic. This one has a change here, here, and here compared to this one. It doesn't necessarily mean it's just one change. So
Gotcha.
that's
Yeah.
what allelic means. It's two sets of instructions that are for the same thing, but they're not identical.
Yeah.
So that kind of leads me to something else in the visual side of it where we're seeing it. So if you've got these alleles and it's purely just a slight change in the instruction, does that kind of give you an indicator of where the actual appearance is coming from? If they don't look the same, like if you take, I mean, there's a lot of allelics that don't look the same, right? Like you take a spot nose and you take a blackhead. They're allelic, but they look nothing alike appearance-wise. You're able to locate those two, so does that give you some kind of a clue or indicator where to look for other things doing something similar, or are they just completely unrelated?
Yeah,
Does
yeah.
that kind of
It
guide
can.
you slightly?
Yeah, it definitely can. With the case of the spider complex, it was really all the work all the breeders did before that. So they knew from breeding that they had to be allelic. And so we knew, oh, we found spider. Well, close to here, we should be able to find spot nose and bongo and, all the things, Wookie. And so that was kind of backwards of what you're saying. But so that can also be the case. If we know from breeding they have to be allelic, then that helps us be able to find the other things that are allelic, like dark matter that we found. The breeder had, bred this kind of funky looking thing to an albino and got that, that dark matter or the dark nova,
Right.
I guess he called it. But the
Dark
reason
Nova
why
in
we were
that
able,
case, yep.
yeah, yeah. The reason we were able to find it is because he knew from breeding it was allelic. But it can also be the other way around. So, like when we're trying to find, monarch, we know it's an albino looking mutation that's not a complete knockout of the melanin. We know, okay, out of these 10 genes that from mice and zebrafish and clawed frog, African clawed frog, all this stuff,
Somewhere
these are
around
like
here.
the 10 genes that make it
Yeah.
look albino. And of those, these 3 or 4 are ones that leave some melanin there or whatever. So yeah, that's definitely a way that original publication on albino and ultramel, all that, she was doing exactly what you just said. She was like, okay, from all these other animals, we know they look like this, and these are the genes that caused it. So let's sequence those genes, and it worked. And that's how we found some of them as well. And that for us, monarch was one of those. We got kind of a general location. We're like, oh, there's this gene right there that causes albino that leaves some melanin. That's probably the one we need to sequence, and that's how we found it.
Gotcha. So Brave's got a question for you directly, Ben. He says, did you go to school for biology and then transition to genetics, or reverse? Were you interested in the genetics first and had to go through the biology route, or how did that work for you in your schooling?
Yeah, good question. For me personally, I knew that I loved animals, and I knew that out of all the subjects that I was aware of, the one that would talk about animals the most was biology, and I purposely avoided every plant class I possibly could and took all animal. Anytime I had a choice, I still had to do some plant stuff. Plants are cool and they're interesting, but I love animals. I wanted as much animal information as I could. So yeah, it was definitely the animals are what brought me to biology. And then once I was there and talking to researchers, I could see the genetics side, the molecular biology side was really interesting to me. Be able to figure out from that, that little bit of DNA code, how that is the instructions for what the animal is going to look like, or what diseases it will or won't have, or whatever. I thought that was really interesting. So yeah, for me, it was definitely biology first and then genetics.
Sweet. Okay. That answers that for sure. And Mitzi thinks you're somehow clairvoyant or something. She said when you brought up mahogany, she had her mahogany out and it was just
Mahoganies are
handling it,
awesome. Sumas
They
are
are.
awesome.
awesome, Gene. That cat
Yep.
just decides on his own when he's going to come and go. So I apologize for Griff there, but yeah, dude, there's so many like weird oddities and things that I have questions about, and I would like to, if you are game for it and you're open for it, could we bring in, like, from our affiliate group a set of questions, or maybe even have you on a Crucible with us and focus on that? Crucible is our affiliate only show where we have affiliates on a panel and we go around robin style where each asks their own question. Would
Yeah.
you be game to be, like, in the smelting
yeah.
pot on that one
Yeah, yeah.
episode?
You'll find if you're talking about reptiles and genetics, I'm very happy to be there. If you start talking about other topics, you might see me kind of like doing this.
Yeah, okay.
But yeah, if we're talking about reptiles and genetics, oh yeah, I would be happy to be there. That'd be fun.
Absolutely. Appreciate that for sure. And we would definitely like to focus a Crucible around that, at least in my mind. I haven't put it out to the rest of the affiliates, but I'm sure they're going to be like, yeah, and the angry horde if I don't do it now. Probably I'll get pitchforked. But yeah, like, okay, so here's one that I know, and it's just on the back of my mind. I got this recently from someone, and it was a little bit of a joking conversation. But if you have that Chimera situation and you were to breed that animal, is there instances where, this is going to sound crude to maybe one person or another, but like one hemipene will pass one part of the Chimera and the other will not? I mean, is there that possibility in genetic transfer from a Chimera animal, or
Yeah,
do
we
we not know
actually
that?
we actually were just talking about that. I was on a show with some boa breeders last Saturday, and they brought up one of the he actually is Matt Shiflett from American Made Exotics. So he does a lot, mostly, I think. Well, shouldn't say mostly. He does a lot of reptile breeding, but I think a big part of it's ball pythons. But he does also have boas. That's why he was watching that show. So Matt has one. We have a video on this too, but it's split perfectly down the middle like this. And this side, it's clearly a desert ghost. This side, it's not. And so in that case, that paradox where it's like just perfectly split, and we tested one side as far as we could away, and then the other side as far away as we could, and one tested homozygous for DG and the other tested het. So they actually asked that same question, and Matt happened to be in the chat. He's like, well, he's an adult now and he would love to breed, but I haven't put him with any girls. So I was like, yeah, if you just get a piece of paper, and if you have someone help you, just have some kind of divider there, pop that and get it from one side, just put a piece of paper and just kind of scrape some of the goo, set it somewhere to dry, pop the other side, scrape it, set it on the other side. Yeah, I would be interested to see too. That it certainly makes sense, unless and from anatomy, those gametes are being produced on two different sides. It's not like they cross sides or whatever. So
That's what I wondered, if they
sense.
shared a transit system, after production transit, or if it mattered which side, or if they're totally separate through the whole process. I don't know the anatomy well enough, but
Yeah, my understanding of the anatomy, it makes perfect sense to me that they would be formed that same way, just like the skin is showing those two different genotypes or two different phenotypes.
Crazy.
I think, and Matt was going to give it a try. So I think if he gives it a try, we'll definitely have to do a video on that if we can actually happen.
That would be
super cool, man.
super interesting, because now how powerful or problematic does that male become?
I mean,
in the wrong situation,
you get to choose, right? You just got
Not
to
really.
tape up one side when you pair them.
Yeah.
Just passing the day.
So
what I-
hemi condoms that blocks one
there you
side
go.
and not the other.
Right.
What I've read, I don't know what all species they've tested it on, but I remember reading where they saw that male snakes did favor one peen over the other. So even though they have two, some males are lefties and some males are righties, and they seem to use that same one either exclusively or close to it. But like I said, I mean, they might have just done that on a rough green snake, or I don't know if that's also that way with ball pythons. But I mean,
Sure.
you certainly could have a vet, clip one side for you if you wanted. But, they could do a surgery and make sure that nothing could come from that other side. But that I kind of doubt anyone's going to go to that extreme. But you never know.
Right. It's just a crazy anomaly that you just got to
It is
consider
pretty
if you
weird.
have that animal. Like this one we have has a lot of things we'd like to use if it was determined that one side was that and the other was not. I mean, obviously, we're not on the level that it's going to make a huge, like, live or die monetary difference, but in our results, it would be super interesting to track and see, right?
Yeah.
Yep. Yeah, and I would love to, since we've had this conversation, we'll see what the panel says. But if, and you said it is a male, right?
Yeah. No, it's it's
I
not
think
male.
this
I
is a
gotta look.
male, I
It's a
believe.
female. No, because
Is it?
we thought it was andro, and andro males don't live, right? Is that correct?
So andro can happen two different ways. Dr. Warren Booth had to explain this to me. We had this conversation within the last 6 or 8 months, but with andro, it's different than partho. Andro can happen in the same way as partho. You can have a single sperm, essentially double, and then you would have the same thing as partho, where you would have it be homozygous. So if it's like a pastel, then you would either get all super pastels or they would just have no pastel at all. You wouldn't get any pastels. But with andro, the other thing that can happen is two sperm can go into one enucleated egg or one egg where there's no DNA there anymore. And
And fill
so,
it up with both
Fills
sides
it
of
with
itself.
both.
yes, so
Right.
you could end
Gotcha.
up with an animal that has it could be male or female. And then it also could be, if it's a python or a boa, if it's a colubrid, that's a different story, or venomous, that's a different story because they have ZW sex chromosomes. But for
Right.
boas and pythons, we have XY. And so andro can give you either males or females, whereas partho can only give you females.
Okay.
Gotcha. Okay.
Yeah, this is not, I'm 90 some percent sure it's female. Just popped it and that's it. Haven't done any probing or anything, but pretty confident it's female.
Okay, yeah, because I was going to say if it's a male and once it's
Yeah.
producing sperm, I would love to also do a panel on a sperm sample and see
Yeah.
if it's exactly the same or how different it is. And it could
Right.
even be a sperm sample one month, you do another one another month, and it could be different if it's, an animal like that.
Yeah, absolutely. Well, if it
Crazy.
comes back positive for everything that was paired up, we definitely would like it to be a male on our end, but I'm like I say, pretty confident it's not. So
So this one's interesting, and I just took it down because it was a lengthier one, kind of pinching the screen. But it's a great question. Genetic deletion. Friends has an Enchi het clown that visually looks super Enchi clown, but couldn't be that. Do you know
Yeah, so
what he's talking
that's
about there?
probably the exact same animal I was talking about earlier. So
Okay.
it tests as Enchi het clown, but it looks like a super Enchi clown. And yeah, that one we've tested. We're okay to swear on this one.
Oh
yeah,
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 no,
you're
no,
good.
you're good.
So we tested the shit out of that one, and yeah, I mean, it just always
Yeah.
comes back Enchi het clown, even though it looks like a super Enchi clown. And I cannot tell you why. That's a super weird. And like I said, that's at least tens of thousands, probably in the hundreds of thousands of clown tests. That's the only time that's happened. How it's doing that, I don't know. That's a
Huh.
good question. That's a weird one.
Well,
Yeah, that I could see that. I mean, a
oh,
long ways out, but I mean, I could see that happening, right?
Yeah, so the way I like to explain this to people is so if you think of like a home pregnancy test, so if you go back to before we had that available, there's so much money that has to go into research and development to produce a test that can be run by a technician that has a lot of experience,
yeah.
and then it's astronomically more expensive to make it
For that next
foolproof.
step, yes,
So
yeah.
if we were doing millions and I would love it if we were millions of reptile genetic tests, all we have to do is if we're selling a million of them and we just put $1 from each test sold towards it, that's $1 million we could use to develop an even more foolproof, simple thing that could be done at home. COVID is a really good example. They eventually got, COVID tests that you could, just spit on it at home and not have to go in or whatever. So that's the problem is the amount of research and development dollars for us to get a test that can be run by a experienced technician is exponentially less expensive than the amount of research dollars that go into developing something that can be done from home. And so you just gotta get out there and get millions more people into reptiles. We get enough people doing tests, then yeah, that and some of that technology may get cheaper and faster and easier. You know, the sequencing the human genome when I was in school in the early 2000s, the US government spent, millions and millions of dollars. There's this huge team. It took them like 5 years. Now I can do it at home with a machine that just fits on a desktop. I could do it in a day or two. And it's going to cost me, two or three or $4,000, whereas they spent, millions and millions. So some of that stuff 5 or 10 years from now may be more doable. But yeah, that's the biggest thing is it's all about volume. If your volume's in the millions and millions, yeah, you can make some really cool, really nice foolproof tests that anyone can do and not mess up and get really good consistent results. But if you're only running, thousands, that's a lot less money you have to go towards research and development.
Right. And so the other thing on that topic really is like you would have to buy off the shelf, I want a spot nose test. Like you couldn't do, hey, check this panel. There's no way that's ever going to be a feasible thing, right? I mean, you'd have to have a way to sort that along. Yeah, I mean, but that bigger issue of exactly what he's saying, like you could do a direct single test, I would think. But if you're going to do like multiples, that's going to be a huge problem.
It would be a lot of work to make it work well for like an end user with no experience running those kinds of tests. Yeah, that would be complicated.
Yeah, just a nightmare.
And
I don't know why
like that for like human diseases or things like that, then it's usually, you spit in a cup and send it in. That's for tests that are millions of them done every year. And like if you're at the doctor, they're drawing a blood sample and sending it somewhere. So, we probably won't ever get there where you could do an at-home panel. That would be crazy.
Right. I don't know why the government spent all that money. All they had to do was just hold a gray down and request the records. Just hold down one of those Anunnaki and go get. Where's our code at, man? Let's have this. Let's forget it. But yeah, no, this is super interesting, and I do see that going that way with everything, kind of to a degree, because that stuff's only going to improve. But where is that initial cost to value return, and you just can't make that work. Not in the small numbers we're talking about. So, yeah. Can you give us a nightmare story? Just one before we let you off the hook here? I mean, no names, just like the worst cluster that you've run into in this whole thing that just even surprised you that you would run into something like this.
I'll have to leave this a little bit vague, but I would say
definitely.
I would say the worst mistake, and this wasn't just one person's fault. It was multiple people discussing how to do this, and but I would say the biggest, probably one of the most expensive mistakes that had to do with genetic testing. And for me, I was lucky enough I wasn't a part of it at all. I was a part of fixing the problem. I wasn't a part of causing it. That's why I'll leave it vague. So
Sure.
Yeah, I mean, the essentially had an animal that was a very expensive animal, had multiple things going on, but it was either asphalt or yellow belly. And the person had very nice high dollar females that had not had asphalt go into it. So he sent it in to get a test done, and the decision was made to run it for yellow belly, and it was negative. And so he's like, oh, then it must be asphalt. Bred it to all those females. Well, for whatever reason, that test was not correct, and so he ended up breeding the yellow belly combo to all his highest dollar females. So that's a pretty big nightmare.
Yeah, especially if it's post sale. That's when it's worse. After they sell all the offspring, if they didn't catch it there. I mean, you would hopefully, but
so stuff happens.
Absolutely, that's bad. Yeah, no names. Obviously, we're going to steer away from that. But
yeah, if you're going to do something like that, it's a higher dollar one. I would never just go off of it was negative for that, so it has to be the other. because it could be stored sperm, it could be this, that, the other. And in that case, it was just that test was wrong.
Yep.
And all other testing on that specific animal and its offspring all was the same thing. But anyway, yeah, I would never go. Me personally, I would have run it for both. And if I was only going to run it for one, I would have ran it for the expensive one and made sure that it was
Yes.
positive for the expensive one.
Right, like run for both only is half the error, because if it shows up negative for both, you gotta rerun it. Something
Well,
is wonky
I think
here, right?
I think
So-
you get a higher odd of a false negative too, right? I mean,
It depends on the test. Some tests have a worse false positive. Some have a worse false
dude.
negative.
Okay.
Yeah. But yeah, I mean, either way, you don't
Yeah.
want to do that.
well, I
The,
think
If it
the other
matters.
the other thing that happened, once again, we'll leave names out, but we had a $20,000 animal that so the person that bred it sold it as being homozygous for a trait. The person that bought it did a panel and it tested heterozygous, and it was a $20,000 animal. So the person, even though it looked like it was homozygous visually, they were worried that the offspring wouldn't test like they're supposed to. All of them should test het from that animal, since it was homozygous. All animals from it should at least have one copy from that animal. So they were worried that they maybe sell a high dollar animal to someone and wouldn't test properly. And so they sent it in again. They said they sent it in, I think, 2 or 3 times. That for sure came from that same animal. Always tested het. I don't know for sure. I was like, I can do some more sequencing. We can try to figure this out. But eventually they're like, you know what? I'm just sending it back to the person that hashed it out. So he sent it back to the person that hashed it out. And he's like, hey, can I just mail this to you? So I got the animal here. It was sent to me. I got the
Oh,
I did a video.
you take the shed.
I took scale clips in case it was like we talked about, a paradox where there's
that's
funny.
patches that test differently. So on the video, so I could send it to both of them, I showed where I took 6 or 8 different scale clips. I ran all of them, and they all said homozygous. So I don't know what happened. I don't feel like either of them were being dishonest, and they both
Yeah.
agreed the animal in the video when I took the scale clips was the same animal that they were both, expecting. It visually looked the same. I don't know for
Wow.
sure what happened. I don't, like I said, I don't feel like either of them was being dishonest. I don't know how,
So-
that mistake could have been made multiple times, but definitely that animal in my hands, all the tests that I did on it all said homozygous. So,
Homozygous, as it
so
should.
went back to good results. Yeah.
yeah, well, I mean, it
Well,
was
good for one party, right?
Yeah.
The other one's
Yeah.
like, what the F? Yeah.
Yeah. Right.
Yeah.
Wow.
So Pat had one too here. I think we jumped over, but there is no blade test, right?
Nope. We would have to get enough sheds come in that people are confident are either blade or super blade, and we just, you don't really see people advertise as such anymore. And I'm also worried that there are either just natural variation or other mutations that aren't the same. If back
It's
when
going to be so
blade-
hard to isolate, is
Yeah.
what you're getting at. Yeah.
Yeah, it's
Okay.
kind of like making the scrambler test also is going to be complicated because you've got, it seems like, popping up in different places. I don't know if there is just one mutation that makes it look like scrambler, or if there's multiple things that give it that look that we're calling scrambler. And if that's the case, for me to be able to design a test is going to be really difficult. So far, I haven't been able to find a clean thing for scrambler, and that may be why. And I think blade would be a similar problem. And we just don't have people sending in blade samples.
Yeah.
So could there be an opportunity for the community to get together as a group and decide that this is going to be our example for a gene? So say you like Scrambler is a perfect example. You've got Scrambler supposedly in a collection. If you test that entire collection and its lineage is passing this from, 3 generations down, and people like what this is passing, they can show a single. Can the community just agree that this is what we're going to call Scrambler and test out from there and just kind of weed out, at least we know that's that line? Or is that, it would have to be kind of a community agreement to say, this is what we're going to go forward calling scramble.
Yeah, yeah. I
right?
mean, to me, that kind of sounds like the guidelines for a dog breed. That's decided
Yeah.
on by a group of breeders. Like, the type of conformation, the ratio of hip height to shoulder height, like all that kind of stuff. Um,
Right.
we definitely could. What I don't know is if there would be a genetic test that would back that up, that would call. I think that would be a visual ID only, at least to start off with. But
Okay.
once you had several animals that fit that description, and we're doing those, kind of registry things we were talking about,
That's why I say,
then
like, get in that
yeah,
collection where he's got multiple
yep,
animals from that he's bred. He's got
yeah,
32 examples, and we can test all those and go, okay, this is the one that no, it's not, same clutch, but at least isolated in that collection. Would that
yeah.
make sense? Or,
Yeah, I mean that would definitely give us something. Be like, yeah, this, these 7 markers that we see in all scramblers to date. Yours has all 7 of those, or yours only has one of those 7, or whatever. I,
gotcha.
I don't
Okay.
know if it would be as perfect as like testing for pied or clown, because
No.
those are so
Right.
easy to know. Yes, that's a clown. No, that's not a clown. That's pretty easy to do. The more difficult it is to know for sure that an animal has a trait or does not, then the more difficult it's going to be to
Proof
find
the
a
test.
marker or markers that we
Yeah.
can do a genetic test for. So, but
Gotcha.
with numbers, if we get enough numbers coming in, then we can solve a lot of problems.
Yeah.
For sure. For sure. All right. Well, we're going to give this the last question of the night, and then we got to get to that damn pop quiz. More realistic for you to offer PCR tests for specific genes if they were willing to get the equipment and knowledge to run it themselves.
So I mean, Dr. Hannah Seidel, Eastern Michigan University, I think she calls it like the Ball Python Project or the Ball Python Genetics Project. She has on her website, she has primer sequences, she has cycling conditions. If it's, I mean, that's honestly, that's how Charlie got started. Was he just saw what she was doing and started talking with her, and so that information is there for some of the mutations in ball pythons. So the biggest downside to it, if you've never done any of it, is obviously the learning curve. So I spent many hours and lots of money on tuition and things like that, to be able to get that experience. You obviously would be, if you're starting completely from scratch, you're going to spend some money. You're going to spend some money on equipment that maybe you end up not liking. You're going to spend money on consumables that, a lot of them are going to go to waste to start off with. It takes a while. Can you do it? Yeah, 100%. Like anyone that's motivated, you want to do it, you want to be able to do it yourself anytime you want. Yeah, I mean, you can make it happen. It's just, do you want to spend that time, or would you rather, pay 30 or $40 to get a couple of things tested here and there? That's the
Yeah.
decision you'd have to make. But yeah, that information is out there for at least some of the morphs for sure.
For sure. And I'm sure a lot of that's like kept as a, well, let's call it a trade secret or whatever you want to call it, but your process is kind of held under lock and key because you invested that time and interest and money
Yeah.
into
I mean,
developing that.
every
And I
year
get
you're
that.
doing something, you'll learn little tricks and little things to make it faster, better, more reliable. I've been doing
Yeah.
this, like I said, since 2002. So I told, we were talking with someone that were, kind of interviewing to potentially work with us. I was like, yeah, the only reason why it's valuable to have me be a part of things is because I've already screwed it up every single way you possibly can. So
Yeah.
if you mess it up and there's a problem, the only reason why it's good to have me around is because I've already done it. So I'd be like, oh yeah, I remember when I did that. This is what
Yep. I
happened.
slipped
You
on
got
that banana.
the chalkboard that's got the
Yeah.
acid off a duck's back crossed out. That one
Oh
doesn't
yeah.
work.
Yeah. So the time and the money has already been spent for
Yeah.
me to know all of the various ways you can screw it up. And if you're starting
Right.
from scratch, you still got to spend that time and money. So this
Yeah.
is just up to you if you want to put the time in. Like I said, that's exactly what Charlie did. He decided he wanted to, and he just kept going and going and going until he could do it and do it for friends. And that's
Yep.
what he did.
Roughnecked it in, and kind of like that, he's said it
Obviously
himself. He
liked
was
it,
just
right?
stubborn-headed, and when he
Yeah.
wanted to learn something, he was going to learn it. It didn't matter if it cost or, And dude, Charlie's an awesome dude. We get along great
Yeah.
with Charlie, and yeah, all of it's possible, right? But like,
Yep.
worth it? Maybe for
Do
Charlie,
you
maybe not for
pay,
me. You know.
do you pay to get your oil changed, or you do it yourself? Do you pay to
Yeah.
get your hair cut, or do you do it yourself? I mean,
Yeah.
there's a lot
Yeah.
of things in life you can do yourself. You just got to decide whether it's worth. Do you cut your own grass, or do you pay someone? Like,
Yeah.
it's one of those
100%.
things.
Now this is
agree.
kind of outside those bounds, and I do think it takes a special person to be able to tailor that and actually have the wherewithal to go through the steps necessary to get it to a point that actually works, right? So
And
Yeah.
also to be able to trust, even if you get a result, you're like, oh,
That's
great, and then do you trust what your result?
exactly.
Like,
That's the big thing right there.
So like
Yeah.
for me, I would be spending money once I finally was able to get results that look like what they're supposed to look. I just don't know if they're right or not. Then I still got to pay for tests for a while and make sure that I always get the same thing that's correct.
right.
Little proficiency testing.
Well,
It takes a
we
while.
appreciate all of you guys that went through that, because I don't wanna. I want to go $30. Please tell me this Camara is not as crazy as it looks. But yeah. So Ben, thank you again, man. We're gonna have you hang out. We're definitely gonna do the pop quiz. Hopefully, people were paying attention. Lost a couple in there because East Coast, whatever gets late. I wanted to run this through to a natural close, and I think we did that. I'm not gonna put you on the hook for it, but we are gonna pass it around the affiliate group. If we could do a crucible with you, I think that's going to work out great. And we'll even
Sure.
structure it like Ben in the middle. We'll have the little lightning bolts kind of coming into the center. We'll try to make that all a thing. But we're
Right
going to
on.
drag Jamie back out. I'm sure out of all that mess, she pulled some kind of a pop quiz for everybody. This is for $10, guys, on the Forge Coin pot. I don't know what she found. Hopefully, it's a good one. It's accurate. We got like 99% results.
It's
on it.
more pertaining to Ben versus all.
Oh, no,
Yeah.
that's fine. Pop
Okay.
quiz for the whole show. It can
Okay.
be about Ben. Yeah.
Yeah. Okay. Pop quiz question. $10 more Forge coin. What year did Dr. Ben obtain his PhD?
Man.
I just won $10.
I failed this horribly. I could guess, but I don't know that I'd be right. I think I know, but I'm not positive.
I remember.
I bet you do.
Me too.
That was a
And
happy
Forge
day. That's
coin?
for dang sure.
Yeah.
Right?
I see what I wrote down as the correct answer.
Yep.
There we go.
2011. Okay, we had E in there a little bit early. We had that.
That
And
was
then
the first
all-
time I sequenced.
Yep. And then we had all
Yeah.
females.
I wrote
coming
that
in.
down too as a question, but didn't pick it.
Mitzi was with E
Oh,
Reptile.
I just
and
messed
then
that up. Sorry.
Right. And then we had Joy too. At 204.
Oh,
So-
sister is here.
Yeah.
Hi.
Hi, Joy.
So we had a lot of guesses, but it looks like we got Connie in there at 2011. Okay. Well, we had a lot of attempts at it. Fantastic. We'll get that Forge coin attributed that way. Ben, thank you again, sir.
Yes,
I know
thank
this is
Yeah, thank you.
something you say you enjoy talking about all the time, but I know you
I do.
also have other things to do. So taking time out of your day to do it, I appreciate that 100%.
You bet.
We all appreciate it.
And you've got a fun community. It's been fun seeing the chat running down the side. That's awesome. Y'all have this going. That's cool.
Yeah.
Yeah, they're a busy
Great.
group, and hopefully they participate, Jay said something. He was trying to. It's these earphones. I apologize. I
No,
talk
you're
over
you're
these
good.
guys.
I was just saying basically the same thing you are. It's a great group. There's always fun in the chat, and just honestly good people that come and watch this. So appreciate them a bunch.
100%. Yeah, Ben, hang out for a minute. We'll talk about how we're going to put you in the Crucible. Everyone else, 2 days left in your week, and you got a weekend ahead of you. We can make it. It's only Thursday and Friday left, and then we got a weekend. We can do whatever the heck we want, right? Guys, thank you for coming out. Next week, we've got HJS coming on.
Yeah.
Rich and Sarah from HJS are going to talk a little bit about how things are going with them with Deeds Not Words and where they're pushing that to. also a little update on their collection. I haven't seen a lot in what they're breeding this year, what they got coming, so it'll be fun to catch up with those guys. Come out and hang with us, no remorse. Ben, thank you so much. Everybody out there, appreciate all of you. As always, have a fantastic week, guys. We'll see you next Wednesday for No Remorse. We'll get away from this Outside the Enclosure thing and do something different. Have a good
Yeah.
night, guys. Peace out.